Hello friends, today we are going to study about bonds involved in stabilizing the secondary structure. Till now, whatever we studied that is about amino acids, peptides, protein sequence, its purification, all comes under protein primary structure. From today, we are starting protein secondary structure. First, we'll deal with the bonds that help in stabilizing the secondary structures. So now, the 3D structure of a protein has three levels. What are those levels? Secondary structure, tertiary structure, and quaternary structure. Secondary structure means it is a local arrangement of polypeptide backbone. How the polypeptides are spatially arranged. That tells the secondary structure. Tertiary structure means it is the structure of entire polypeptide which includes the side chains. Secondary structure is only the polypeptide backbone. Tertiary structure is the 3D structure that includes entire polypeptide, that is the backbone as well as their side chain. Quaternary structure is the arrangement of protein subunits. That means it can consist of more than one polypeptide also. So these are the levels of protein 3D structure: secondary, tertiary, and quaternary structure. Now, 3D structure of the protein is determined by its amino acid sequence, which we already studied, and the function of the protein depends on its structure. Now, any isolated protein can exist in one or a small number of stable structural forms. And these structures are maintained by non-covalent interactions. If we see the primary structure of a protein, what is the primary structure of a protein? It is a sequence and composition of amino acids. What is the bond that connects these amino acids? Peptide bond. What kind of bond is this? It is a covalent bond. Whenever this primary structure gets folds into 3D structure, secondary, tertiary, and quaternary, the main stabilizing interactions are the non-covalent interactions. What are these non-covalent interactions? These also we studied individually about these interactions in different videos. Hydrogen bond, hydrophobic interaction, electrostatic interaction, van der Waals forces. We already studied about these interactions. These are non-covalent interactions that mainly help in stabilizing the 3D structure of any biomolecules, be it a protein, DNA, carbohydrate, lipids. Now, all these interactions are found in all biomolecules. Hydrogen bond, where you will find hydrogen bond? Wherever two high electronegative atom is bridged by hydrogen atom. In all those places where two electronegative atom is bridged by hydrogen bond, you'll get a hydrogen bond formation. Hydrophobic interaction, whenever any molecule wants to separate itself from the surrounding aquatic environment, there you can find hydrophobic interaction, mainly in the interior of protein or DNA in any biomolecule. Wherever the biomolecules will aggregate, in the center you will find the hydrophobic interaction responsible. Electrostatic interaction, where you can find electrostatic interaction? Wherever in the biomolecule there is the opposite charge come in close contact with each other, you'll find one electrostatic interaction will take place. Van der Waals forces, when the molecules are close enough, this force comes into play. So all these force you'll find in all the biomolecules, be it a protein, carbohydrate, lipids, glycoprotein, glycolipid, everywhere. And the primary structure of all the biomolecules are stabilized by covalent interaction. Now, come to protein structure. Conformation. What the word conformation means? It is the arrangement of atoms in a protein. Now, the conformation includes any structural state that can be achieved without breaking covalent bonds. For example, in protein, we have NHCO peptide linkage. So, this type of conformation is known as trans because here the H and this O are in opposite direction. This is a trans. It can be in cis form also. So these are conformations. That means the rotation around single bond. During this rotation, whether it is a trans conformation or whether it is a cis conformation, it is the rotation around single bond, but during this transformation, this bond is not hampered. So conformation means any structural state that can be achieved without breaking covalent bonds. For example, rotation around single bond. Now, few conformations are stable in biological conditions. And which conformation will be stable? The ones that are thermodynamically favorable or having the lowest Gibbs free energy. The relation of stability with lowest Gibbs free energy we already studied in the thermodynamic VD. Now, what are native proteins? The proteins that are folded in a way that they are functional in nature is known as native proteins. Now, stability of protein conformation. What do we mean by stability? Stability means tendency to maintain a native conformation, that is, the condition or the conformation in which the protein is stable and in a folded form and in a functional form. So, during this, the delta G should be minimum. So delta G separating the folded and unfolded status differences. In folded, it is 20 kilojoule per mole. In unfolded state, the energy raised to 65 kilojoule per mole. Now, unfolded state will have high entropy. So whenever there is high entropy with hydrogen bonding interaction, that combination favors the unfolded state. What favors native conformation or folded and functional state? The disulfide bond and the non-covalent interactions. Now, when we say that non-covalent interactions are stabilizing the biomolecule, but these non-covalent interactions are the weak interactions, then how come they can help in stabilizing these biomolecules? Because they are present in multiple numbers. Due to its involvement in maximum number, they help in stabilizing the biomolecules. This is again the same thing. In general, the protein conformation with the lowest free energy is the one having the maximum number of weak interactions, because these weak interactions are only responsible for stabilizing the 3D structure of a protein. And what are these weak interactions? Hydrogen bond, electrostatic interaction, van der Waals forces, and hydrophobic interactions. Now, this first one is the protein in unfolded state, and the second one is the protein in folded state. Now, what is happening here? When the protein is in unfolded state, because the cell is 70% aquatic, so surrounding water molecules are present. So when the protein is in unfolded state, these water molecules are interacting with this unfolded protein through hydrogen bond formation. So the water molecules are now forming the bond with unfolded protein structure. In the absence of this protein, the water molecules are bonded with each other. But as soon as the protein is present in the aquatic environment, now this bond will break and this water molecule will go and interact with this unfolded protein. So here, bond formation and bond breakage, both the phenomena are happening. But because more of water molecules are now involved in bond formation with this particular unfolded protein, therefore less water molecules are available freely. So that means what is happening here? Entropy decreased. The entropy of this universe decreased. What happened in the folded state? As soon as the protein fold, that means now the bond which was there earlier with water molecule, that bond will break open and the protein will form bond with itself. The amino acid present here is forming the bond with the amino acid present here, which was earlier involved in the bond formation with the water molecule in an unfolded state. Now more water molecules are again become free. So that means the entropy increased here. In the folded state, entropy increased here. More of entropy is thermodynamically favorable according to the second law of thermodynamics. The entropy of the universe, that is system plus surrounding, will go on increasing. Come to the enthalpy, the first law of thermodynamics. What is enthalpy? The free energy content or the energy content. Now, here the bond formation and bond breakage is also happening. In the folded state also, bond formation and bond breakage both is happening. More energy is required to form a bond than to break a bond. Hence, here more bond formation is happening between water molecule and the unfolded protein. Therefore, here also delta H is negative. But in the folded state, the delta H is positive. This also we already studied in the previous videos of hydrophobic interaction as well as during thermodynamics video. Those who have not seen those videos, I would request you to check those videos once. Now come to the hydrophobic interaction. Now again, the same thing which I just now explained in the last slide, as well as the same diagram and explanation is there in the hydrophobic interaction. Correct? What happens whenever any non-polar substance is added in the aqueous environment? What will happen? The water molecule, which was earlier bonded among themselves, this bond will break and this water molecule will start forming the bond with the non-polar substance. So what is happening here? Delta H negative and delta S, that is entropy, is also negative because here the randomness decreased because more of water molecules are involved in bond formation. And the non-polar substance also cannot move freely because it is surrounded by water molecule. So delta H is negative, delta S is negative. Here delta G, that is Gibbs free energy, becomes positive and the reaction is non-spontaneous. After some time, in due course of time, these non-polar substances will aggregate. So when they aggregate, now less number of water molecules are required to surround them. So more water molecules are free outside. So when they are free outside, here entropy increased. Similarly, enthalpy also increased because more energy is required to form a bond than to break a bond. Delta G, that is Gibbs free energy, becomes negative, and when it is negative, the reaction is spontaneous, that is thermodynamically favorable. So this is the reason why protein folds, because there is a tendency in non-polar groups to form a cluster. Therefore, folding of protein is favorable thermodynamically. For the detailed explanation of thermodynamics, you can check the thermodynamic video. The hydrophobic effect, which causes the non-polar substances to minimize their contact with water, is the major determinant of native protein structure. As I said earlier, that all the non-covalent interaction will help in stabilizing all the biomolecules. But some non-covalent interactions will play a major role compared to others in stabilizing some particular biomolecules. For example, in protein, a major non-covalent interaction that helps in stabilizing the protein is hydrophobic interaction. When we will study DNA, we'll found that the major non-covalent interaction that helps in stabilizing the DNA is hydrogen bond. Though all the non-covalent interactions will be found in protein also, all the non-covalent interactions will be found in DNA also. Now, what is hydropathies? This is the combined hydrophobic and hydrophilic tendency of individual amino acid residues in proteins. This is the list of amino acids with their hydropathic nature. Like, for example, isoleucine. So, isoleucine is much more hydropathic compared to valine. Valine is 4.2, leucine next highest. This is the list of the amino acid with their hydropathic nature. Now come to hydrogen bond and ionic interactions. Now these also follow the same entropic effect. Like when they are involved in bond formation means more of water molecule outside are freely available. That means increase in entropy and hence it is thermodynamically favorable. The bond formation is thermodynamically favorable. Hydrophobic residues, these are buried inside, and during this, whenever two electronegative atom is bridged by hydrogen, gets involved in hydrogen bond formation and helps in stability of the protein. Van der Waals forces also plays an important role in stabilizing the protein. Now next is a disulfide bond. Disulfide bond is a covalent interaction that helps to fold the protein in its native state. This bond is rare in intracellular protein because the environment of cytoplasm is a reducing environment. What happens if the environment is reducing? Disulfide bond is a bond between two sulfur present in cysteine in the reducing environment. What do we mean by reducing environment? More of H plus. When more H plus is present in the surrounding, this bond will break because they will get converted into sulfide group. During protein sequencing also, we studied that we can cleave the disulfide bond with the help of a reducing agent. So because in the cytoplasm, the environment is a reducing agent, that is more of H plus. Therefore, in intracellular proteins, the disulfide bond presence is very rare. Mainly, the bonds are present in the proteins that are secreted outside the cell because outside environment is more of oxidative in nature. Hence, disulfide bonds help in stabilizing the proteins that are secreted outside the cell. Clear? Coming to metal ions, that usually helps in cross-linking of protein. For example, zinc fingers. There are many enzymes that work efficiently in association with the cofactors, that is metal ions like zinc, magnesium, manganese. So what these metal ions do, they help the protein or the enzyme to interact with the DNA. So it helps in stabilizing also, as well as it helps in interaction with the DNA molecules. Now one question from my end for you all, that you have to tell me which bond is responsible for the stabilization of primary structure. Do let me know in the comment section. If you found the video informative, please do like, share, and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.